So thank you, sir. Thank you for taking out time for us, and I'll be participants. So we have Dr. S. Mohan, and he is he's working as a scientist at CSIR Indian Institute of Chemical Technology, and he since 1998 he has done his BTech in civil engineering, MTech in environmental engineering, and PhD from Sri Venkateswara University, Tirupati. Dr. Mohan was visiting professor at Kyoto University in 2005. He is also an Alexander Humboldt Fellow at Technical University of Munich, Germany from 2001 to 2002. He was a international fellow at South Korea in 2018. Dr. Mohan is placed in second position in India and 29th position in world in the field of biotechnology by Stanford University study on 2% global scientist list. Dr. Mohan's research majorly intends to understand and respond to human-induced environmental changes in the framework of sustainability in the interface of environment and bioengineering. His research focuses on designing self-sustainable systems and technologies through a nexus approach, enabling waste as a feedstock towards the production of low-carbon energy, chemicals, materials, adopting regenerative resource management. His research interests are in the area of green chemicals and fuels, acidogenesis, microelectrogenesis, photosynthesis, carbon dioxide sequestration, circular bioeconomy, waste remediation, ecological restoration, and sustainable engineering. He also works on various industrial and societal consultancy projects. He demonstrated the production of green biohydrogen with simultaneous waste remediation and established a first of its kind of waste-fed biorefinery platform at the pilot scale. Dr. Mohan has authored more than 420 research articles, 63 chapters of books, edited 5 books, and has 9 patents to his credit. His publication had more than 27,000 citations with an H-index of 88. He has guided 37 PhDs, 2 MPhils, and more than 100 MTech and BTech students. Dr. Mohan is recipient of the coveted Shanti Swarup Bhatnagar Prize for the year 2014 in Engineering Sciences from Government of India. He also received several awards and honors, which include Abdul Kalam Technology Innovation National Fellow by DST SIR and INAE in 2022, Golden Peacock Eco Innovation Award for the year 2022 by Institute of Directors, Dr. C.V. Raman Award in 2020 by Government of Andhra Pradesh, DBT Tata Innovation Fellow of 2018 by Department of Biotechnology, Vaishvik Award for the year 2018 in the category of Environmental Science and Technology by Vividakshi Ayodhik Samsodhan Vikas Kendra, Most Outstanding Researcher in the Field of Environmental Science in India 2018 by Careers 360, National Bioscience Award in 2012 by Department of Biotechnology, SIR IGCW 2017 for Biohydrogen Technology from DST SIR, Environmental Engineering Design Award in 2017 by National Design and Research Forum of Institute of Engineers in India, Prosper.net Scopus Young Researcher Award in Sustainable Development in 2010 under the Energy category by United Nations University, and Elsevier NSI Scopus Young Scientist Award in 2010 in Earth, Oceanographic and Environmental Sciences by NSI and Elsevier. Nawab Jain Jang Bahadur Memorial Prize in 1994 by the Institute of Engineers, etc. So Dr. Mohan is elected Fellow of National Academy of Engineering, Biotech Research Society of India, Telangana, Andhra Pradesh, Academy of Sciences, International Forum on Industrial Bioprocesses, Institution of Engineers, International Society for Energy, Environment and Sustainability. Dr. Mohan is serving on editorial board of journals like Bioresource Technology, Transactions of the Indian National Academy of Engineering, Environmental Technology, Material Circular Economy, Carbon Resources Conversion, Journal of Environmental Science and Engineering, Material Science for Energy Technologies. So with this introduction, I would now like to request Dr. Mohan to start with his presentation. Indeed, a very impressive CV. We are very honored to have you, sir, today with us. And I hope all our participants will enjoy your talk and learn a great deal from your work. Thank you, sir.
Thank you very much, Dr. Amy, for a nice introduction. Hope I have about 20 minutes time for presentation. No, audio. Dr. Annie, I am not.
Yes, sir. Your duration is 30 minutes, followed by question and answer.
Okay, okay. Thank you very much. Thank you.
Yes, thank you.
Very good morning to everyone online as well as offline. It's my pleasure to speak to you today on some of the work which we are doing in IICT. Specifically in this presentation, I am going to discuss about biorefinery concept with respect to waste as a feedstock in circular mode approach. So first I would like to thank organizers of this international conference, NIT Jalandhar, as well as Dr. Nikhil, who is also part of some of the work which I am presenting here. So thank you very much for the opportunity and platform. So I'm from Indian Institute of Chemical Technology, which is a premier laboratory under Council for Scientific and Industrial Research. Our department specifically works on various technologies related to environmental engineering, and apart from that, we are also looks onto bioengineering aspects, specifically focusing on using waste as a feedstock, which includes solid waste, liquid as well as gaseous waste, and try to use biological routes as well as biocatalyst, and then look into production of some chemicals in the form of fatty acids, polymers, fuels in the form of biohydrogen as well as HCNG. So our approach basically looks into the circular economy. Resource efficiency is our major interest, like to loop the systems into closed loop systems, and also try to understand the process with using life cycle analysis. Some of the work I will present in this presentation. So to start with, I would like to put this slide. We have to, with the present concern with climate issues, everybody know that there are a lot of challenges. And apart from challenges to attain sustainability, there are opportunities also towards moving to net zero pathway. And that requires strategies as well as interventions. Very critical requirement is there. Try to put some of the requirements as such, it's not requirements, such strategies that should help to achieve the net zero pathway, which is being anticipated. So the first one, decarbonizing process, is very important to achieve net zero emissions, which will have direct influence on reduction of greenhouse gas emissions. So this is very critical issue. And apart from that, replacing the fossils with renewables, particularly by using green low-cost chemicals or low-cost fuels. Particularly in this presentation, I will be presenting on green hydrogen. It's very important, and also going towards semi-synthesis by using biomass as a feedstock. All these things, finding low-cost feedstocks alternatives, will have a direct role to play on the sustainability issues. As I told, infusing renewables in energy as well as material domains is important, and energy efficiency, which energy losses, if we can tackle, that will also have direct role to play, apart from waste management and carbon capture utilization as well as storage. And more important, moving towards circular economy to reduce our requirement of materials. That's primary material requirement is also very importantly required for the coming days, but to adapt into different kind of systems, not only for waste remediation or energy, but the circular economy will play a major role in many things. So, and adapting nature-based solutions, as you know, AR6 report has specifically mentioned that nature-based solutions as well as circular economy have direct role to play on sustainability. So these two are also very much important. And as you know, individual responsibility will always have a direct role to play on sustainability. Everyone can contribute their role towards attaining sustainability. So keeping all this view in this presentation, I will focus on biorefinery, but we'll start with green hydrogen production. Everybody knows now that green hydrogen is talked much, particularly with respect to electrolysis, which is termed as green hydrogen at present. But in some of the definitions, biological routes of hydrogen production are also considered as green. So there are a number of colors applied to hydrogen, gray, brown, blue, green, yellow, like that. But we will be looking into these aspects. Electrolysis is not our domain, but we'll talk about some of the biological routes, how we can produce hydrogen. So normally, green hydrogen, there is a standard definition which is coming in global thing that 1 kg of CO2 produced from the process of producing 1 kg of hydrogen is termed as green. So still we have to achieve throughout the globe, there is significant efforts made in towards reducing the emissions during process. So then this definition will apply as a green. So that's why instead of fossil-based energy for electrolysis, government is trying to induce solar-based energy so that significantly CO2 emissions can be reduced while producing hydrogen. So there is significant reduction, about 60%. So we started working on biohydrogen production from 2005. Our basic interest is to valorize resources from waste. When we go to directly anaerobic process, we will be losing materials in between, but we will get methane, which is a very important gas at present being used with the existing infrastructure. But our interest is to regulate the metabolic activity towards some terminal products, which is of importance. So basically, when we look into hydrogen production, there are two classifications. One classification I would like to say based on the feedstock. So when we use biomass, then it is thermochemical route. We can produce hydrogen, which can be termed as biohydrogen because it is coming from biological resource. The second classification, it is process based, where biological process can be subdivided into basically for four things at present. One is anaerobic, which everybody knows it's a dark fermentation, and a matured process at present, which is up to TRL6 level and can have chances to scale up. The other process, photobiological, theoretically this can need higher hydrogen, wherein anaerobic process we get 4 moles of hydrogen with 1 mole of glucose. But photobiological process can produce 8 moles of hydrogen with 1 mole of glucose. But there are inherent disadvantages with respect to the production of oxygen and heat entrapment. Many issues are there. So practically scaling up, still there are many limitations. But we can integrate dark fermentation as well as photobiological process in order to increase the productivity rates. So third process is based on the enzyme, which is producing hydrogen, that is hydrogenase enzyme. That work is being done at a relatively less phase, but still at the laboratory scale. The fourth important process, which is similar to microbial fuel cell and use same mechanism as anaerobic dark fermentation process, but with induced electrodes, can produce hydrogen. This is also one of the process which is being studied at present in a very significant way. So as I told that we started working on 2005 onwards on hydrogen production. Dr. Nikhil also contributed in some of these aspects to go towards pilot scale, wherein in 2016 we are able to set up pilot plant with 10 meter cube capacity with the funding of MNRE. And then we have developed a waste biorefinery platform in order to use the waste coming from hydrogen reactor, so that increase the resource efficiency. In 2007, with the funding of CSIR, we have operated waste fed refinery in a closed loop approach, which I will present in this thing. So coming to hydrogen production through dark fermentation, it's very much similar to methane production, where in methane production, you know, there are 4 steps, basically hydrolysis, estrogenesis, as well as methanogenesis. But in this process, we have to terminate methanogenic process to make estrogenesis as a terminal process, wherein hydrogen, CO2 and acetic acid will be produced. So the success of this process will depend on how effectively we are terminating methanogenic activity. Otherwise, it will go to methane as a terminal end product. So when we look into the productivity based on the theoretical limits, 1 mole of glucose, when it goes to acetic acid pathway, we will get 4 moles of hydrogen, or 1 mole of glucose goes to butyric acid pathway, we will get 2 moles of hydrogen. In COD terms, 1 kg COD, we are able to remove, we'll be able to produce near to 450 liters of hydrogen. This is the theoretical figures, but we are only getting 50% of the theoretical, which because of the inherent problems existing with acidogenic process. So we started with different kinds of wastewater, using different kinds of wastewater to produce hydrogen by dark fermentation process, and reasonably good hydrogen production was observed. And again, it depends on the amount of carbon load present in wastewater, as well as the nature of wastewater, biodegradability, with reasonably good CO2 removal efficiency. This is also again very important when we look into the wastewater treatment. So we observed to scale up, there are many limitations, because which we need to resolve these limitations, unless otherwise we cannot scale up the process. Some of them are listed here, biocatalyst based, because system is using mixed culture, there will be always synergy action of methylogenic bacteria, which will try to convert H2CO2 and fat acids into methane. That is not our requirement. And feedstock, availability of feedstock, organic loading rate, biodegradability, and solid waste if you are using pretreatability, and supply chain are major issues with feedstock. So coming to process, feedback inhibition due to formation of fat acids is a major issue. So apart from pH drop and buffering, low substrate conversions are major limitations. The fourth one, outlet from the fermentation system, which is producing hydrogen, have still 50% of carbon in the form of fat acids. So that needs to be treated before disposal. Apart from that, there are economic, safety, purification, other issues apart from policy that is not part of this presentation. I would like to briefly explain some of the things how we try to resolve before scaling up to pilot scale. The first one is how to regulate the metabolic activity. So we try to use classic microbiology techniques, wherein by giving shock loads, we'll be able to eliminate non-methylogenic bacteria, which is methane bacteria, basically. So at extreme conditions like a high acid condition, alkaline or heat or oxygen shock condition, there will be reduction, there will be inhibition of the methylogenic bacteria, which are non-spore forming bacteria. But in case of hydrogen producing bacteria, which are mostly formicutes, are spore forming bacteria, wherein after applying this pre-treatment, they will go into spore formation. And then subsequently we can enrich and then use for hydrogen production. So this has helped us to increase the hydrogen productivity when compared to control, which is having methylogenic bacteria. So there is significant increment in all the conditions, and applying this treatment every day we can do, not apply in the same reactor. So we try to use, we try to operate different systems with almost 1 year of time by giving different pre-treatment strategies in order to understand the process efficiency or process dynamics and as well as microbial diversity. So this shows that even after operation of 250 cycles, we have only given 2 times pre-treatment in between. That is somewhere between 4 months or 3.5 months. And subsequently at the end of the operation, when we look into the dynamics, microbial diversity analysis, it shows that there is very specific enrichment of formicutes in all the pre-treatment strategies, where you can see the blue one here, wherein the formicutes was the major contributors here. So there is no problem with respect to maintaining of culture when we operate system for a long time. When compared to untreated inoculum, there are more diversity, phylum diversity, you can see here in the first left side figure. So then the reactor configuration is very critical issue, because the system is always moving towards acidogenic in nature, and feedback inhibition is a major issue. So once we give robustness to bacteria which is in the system, that will have a lot of advantage. So when we compare with suspended growth system and biofilm system, wherein the biofilm system has showed significant advantage, that is particularly with respect to productivity or persistence of the bacterial culture. As you know that biofilm systems always try to form biofilm on a substrate. Normally the biofilm formed at the surface will have a lot of advantage, robustness, compared to the bacteria which is in form of suspension. So that advantage gave us the higher productivity. So compared to suspended, biofilm are more efficient to sustain the dynamic nature of the system microenvironment, as well as to maintain the sustainability. So other important issue is organic loading. When we are looking into the productivity and the scaling of operation, the higher the hydrogen production, will be able to get a reduced cost of hydrogen. So we try to look into the increment of COD loading rate, particularly from starting with 6 grams of COD per liter, and we try to increment it up to 50 grams COD per liter, wherein the maximum productivity was observed. And subsequently, we increase the organic loading rate from 50 grams COD per liter. There is drastic drop in hydrogen production, particularly up from 66 grams per COD per liter when we increment. And there is, you can see the pH graph where this crossed 4, reduced from 4, which inhibit the system performance because of deactivation of bacteria. So buffering plays very major role here. So we try to use some of the buffering inputs in order to increase the buffering capacity of the system so that it can increase the productivity. So by giving some strategies, we are able to again regain the performance, where you can see, and pH also came to 4.88. So this help with the buffering of CO2 in some cases, and very low cost inorganic ions. By giving buffering, we'll be again retain the persistent performance or operating at pH 7. So these strategies keeping when we look into the diversity of the long-term operation, all the systems, pilot plus lab scale systems we have operated, we observed firmicutes will have a major role to play, and firmicutes are both anoxic as well as anaerobic nature persistent, such as bacillus. So these are various bioreactors which we have used in laboratories, starting from small 1 liter capacity to 20 liters capacity, semi-pilot scale system. And based on this, these are some of the optimum conditions which we try to get from the experiments which we have done. Based on this, we try to scale up the system. This is a 10 meter cube pilot scale system, reactor capacity, which can produce about 50,000 liters of hydrogen per day. This was funded by Ministry of New and Renewable Energy. So the P&I diagram was depicted in this figure, wherein you can see all the reactors and other accessories which has been integrated with main hydrogen reactor, which is a biofilm system, as you can see here. There are pre-treatment system and as well as buffering control systems, as well as gas holding and pre-treatment of biocatalyst. All that unit operations are there. And these are the photographs where you can see the 3 storage pilot plant system with the different accessories. So the most of the thing have to go with gravity. We try to reduce the usage of pumps so that we can reduce input of energy into the system. So we try to use basically biogenic waste, food waste, at 50 grams per COD per liter of organic loading rate. We are able to get 50,000 liters of hydrogen. So here, this is shown in one of the cycle operation where you can observe more than 50,000 liters of hydrogen per 2 days. And here you can also see that enrichment of firmicutes here, very specifically compared to parent inoculum. You have been you can observe enrichment of firmicutes at the end of the pilot scale operation. So these are some cost calculations we have done initially. The 10 meter cube capacity, the hydrogen production rate, we are able to get hydrogen at a cost of 240 rupees per kg. But if you are able to increase the productivity plant capacity, we can reduce hydrogen production of the from the system. So green hydrogen production. So based on this, we also understood that there is significant scope to reduce the cost of hydrogen apart from recovering the resources. As I told, that outlet of hydrogen reactor still contain 50% of carbon load. So this biorefinery strategies we try to understand, wherein Dr. Nikhil also worked in many of these aspects in our lab. So hydrogen outlet of hydrogen reactor, there is will contain a significant concentration of fatty acids in the form of acetic, butyric, and propionate. So when we integrate with conventional anaerobic process, these fatty acids will convert into methane. So we get two forms of energy apart from 80 to 90% of resource recovery. In a similar way, when we integrate with microbial electrolysis cell by applying the certain potential, it can generate hydrogen apart from wastewater treatment. And another important thing is when we use this as a feedstock for bioplastic production, there is a very good productivity of polyhydroxyalkylate, which is a biopolymer. Or directly after dilution, this can use for farming activity. Because of fat acids and mild acidic nature, the phosphate availability for the roots can be augmented. So that will help to produce, that will have direct role on plant productivity, which we observed. We have operated these bottle-ground plants for almost 1 year and to assess the data. And another important integration I would like to say with mixotrophic algal cultivation, the fat acids, when subjected to microalgae cultivation, will be able to convert the carbon into biomass as well as lipids and other fractions, apart from 90% of CO2 reductions. And in microbial fuel cell integration, we get additional electricity or photofermentation, as I told earlier. This integration will give additional hydrogen. However, sustaining this for quite a long time is very difficult. Still, we are trying to look into these aspects in order to get higher productivity. So all this integration, I would like to say, it's a kind of biorefinery integrated strategy, will help us to understand the possibility of integration towards resource recovery and designing the waste bed biorefineries, which I will present at end of the presentation. So as I told earlier, integration with methanolytic process, we will be able to produce both hydrogen and CH4, wherein at present supplementing of less than 80%, 18% of hydrogen in compressed natural gas is being proposed by government of India, which will have direct advantage, wherein it can use the same existing infrastructure and reduce emissions and increase the engine efficiency also, which is termed as HCNG, hydrogen compressed natural gas, or in some terms called as ethane or bioethane. As I told earlier, fat acid will be, by anoxic conditions, can be converted into plastics, polyhydroxyalkylate, or for organic farming, for phosphate, as a phosphate solubilizing agent. A significant productivity was observed with all kinds of, with the leaves, number of leaves, number of flowers, as well as bottle bag numbers also. There is significant increment. So in another integration, wherein this is recently we are pursuing, the fat acid, which is coming from hydrogen reactor, they are very good platform chemicals. As you know that acetic, nitric and propionic acid at present is being produced from fossils and have wider applications. So when we are trying to produce these things, why want to waste it? So we try to focus on increasing the productivity of acetic as well as nitric, propionic acid, wherein theoretical terms 1 kg of COD will be able to produce 0.62 kg of EFA when it goes to acetic acid pathway, or 0.41 kg of EFA when it goes to nitric acid pathway, or 0.65 kg of EFA in propionic acid pathway. But this pathway consumes hydrogen. So for hydrogen, this is not beneficial, but these two will simultaneously produce hydrogen, a win-win situation here. So we're trying to understand the mechanism and productivities. Our basic scope is to increase the productivity rates in order to get to 6% of the liquid volume, so that separation will be easy. So increasing productivity also increase the hydrogen productivity. So we can reduce the cost of hydrogen. So these are some of the data consolidated, which we are working with the different conditions and different streams to increase the productivity of fat acids, which I will not explain. So still we are working on this towards increasing the productivity more than 5%, so that recovery will be more beneficial. So before closing, I would like to show this slide. This is the slide which try to integrate, starting with acidogenesis. I would like to say anaerobic bacteria are very versatile. At the starting phase of any biorefinery, try to reduce complexity of wastewater and try to produce some of the platform chemicals or fuels that have direct important role or direct role to play in biorefineries. for multi-product requirements. So try to look into different integrations where it can go, starting with waste and then sugars and fatty acids, and then for different products. So this kind of things have a very beneficial effect on sustainability, which needs to be addressed by the scientific community. So the second process I'm very much interested in biorefineries is integration of bioelectrochemical systems, a kind of microbial fuel cells for value addition. So this is very much similar to anaerobic process, as I can say, like our acidogenic process where we are producing hydrogen. Same culture will be used in a defined system where electrodes are there. So by inducing the electrode system at certain potential, particularly in this case minus 0.6, we'll be able to gain higher hydrogen productivity with less COD production. So this will also help to use the fat acids. Our interest is to use fat acids on first stage of acidogenic process as a feedstock for microbial electrolysis, where we can produce additional hydrogen. So this has gave us a very good leads, and we try to focus on understanding what is happening with the bacterial metabolic activity. One of our PDF, she tried to use E. coli and look into the pyruvate metabolic pathway, try to study some of gene expressions which are critical in producing different kinds of fatty acids as well as hydrogen production. So in these figures, you can see that there is direct role to play different potentials. The productivities are different. At the same time, the enzyme gene expression levels are also very diverse. So this shows that we can regulate, or we can, by altering the potentials, so we can get the product which is of our interest. So this shows us interest, and now we are trying to extend from E. coli to Bacillus subtilis to understand with more gene expressions which are of our interest. In this study, the metabolic flux analysis with electrogenic thing, we observed that there is possibility of succinic acid production at particular potential, which is high value products and precursors for many chemical reactions, including plastics. So this is the gene expression levels at different potential. You can see there is very diverse variations in the expression level. So still we are trying to look into this to understand how can we regulate metabolic activity. And based on that, parallelly, we also started working on fermentation in a defined electrode system, electrode based system, by applying potential, applied potential of 0.6 and closed circuit, specifically focusing on fat acids, where we are able to increment by 20 to 30% of productivity. Fat acids as well as relative abundance of hydrogen was also observed. But interestingly, diversity, unlike acidogenic process, here probiotics showed a higher increment in the operation of electrofermentation system. However, fat acids are also being produced. So then our focus in a refinery, as you know, that carbon capture and utilization also plays important role, and along with hydrogen, there is possibility of, not possibility, 50% of gas composition contains CO2. So when we look into the net energy CO2 production, we have to significantly reduce here also, even though this is biogenic in nature. So we try to focus on using CO2 as feedstock by a specific group of bacteria, homocytosolic bacteria. So we are trying to use two kinds of system. One is gas fermentation system with high pressure, and microbial electrolysis system. In gas fermentation system for wood-lingual pathway, hydrogen is required to convert CO2 by homocytosolic bacteria to fatty acids. But in electrochemical system, that is microbial fuel cell kind of system, there is no need for giving hydrogen, but external potential electron flux will help to divert the reaction towards acetic acid production by directly cycling CO2. This is a high pressure gas fermentation system fabricated in laboratory, which can go to 5 bar of pressure. We have operated this with 2 bar. So gas fermentation system is able to give about 3.9 grams per liter of fatty acid, wherein the conversion carbon, inorganic carbon conversion rates are about 16.6 mg per liter. So when we compared with microbial electrolysis cell with dual chamber, the productivity are able to increase without giving hydrogen additionally. And also at the same time, you can see the succinic acid production also was incremented. So our focus is now also on producing succinic acid by biological route by both adapting gas as well as the electrochemical system by taking into advantage of reverse TCA cycle, wherein hydrogen is also important electron donor. So we are trying to look into hydrogen as electron donor as well as electron flux also in place of hydrogen by taking into advantage of reverse TCA cycle with our specific ITLS. One of the PhD student isolated citrobacter amino acid. So keeping all these things, I try to present at the before closing of the this talk a refinery concept, which is the title of this presentation. As you know, that refinery is basically which takes crude from Mother Earth, which is basically chemical in nature, and try to refine it in a refineries to different kinds of products. So in a similar way, here we are trying to look into a biorefinery wherein biological feedstock or biological process is used by taking waste as a bycrude, as a waste as a crude, very much similar to fossil-based refineries. Try to design a closed loop approach. This is the waste biorefinery model, which we have developed in IICT with the funding of CSIR by integrating with hydrogen system, which is given by MNR. The basic objective of this is to look into the integration of various biological processes, not to make resource recovery more efficient. So for try to explain this one, we can take wastewater or waste. If you are taking biogenic waste, we have to pre-treat it. And initially, this will go to microbial fuel cell kind of system, wherein to simplify the complexity of wastewater and to make the carbon amenable for bacteria in secondary process. Here we will be able to generate a little bit of electricity if we want, or otherwise we can induce in a closed circuit mode operation where we can still reduce the complexity of wastewater. So then subsequently the waste pre-treated water will go to the acetylogenic reactor, wherein we will be able to produce hydrogen. 60% of carbon will be converted here into hydrogen, and then the outlet will have rich in organic carbon. We'll go for any of this process for photohydrogen, for hydrogen production, or methanogenesis for methane production, or enoxygenesis for bioplastic, as I explained earlier. By way of three-stage integration, we will get three forms of energies or two forms of energy on biomaterial here. Like so, 90% of carbon, near to 80 to 90%, we will be able to convert, and the residual carbon will be treated in wetland systems and can be used in process when we are using waste for dilution, the closed loop approach, or we will be able to send it for gardening, which is very sustainable. So by way of this, we can increase the resource recovery as much as possible. We can also close the loops by not releasing into the environment. So this we try to construct. This is a top view of waste biorefinery facility with different anaerobic digesters, algal cultivation system, wetland system, bioplastic systems, different systems. And this is the closed view of different unit operations. So this when we present into a consolidated data, standalone and integrated biorefinery system. In standalone system, we'll be able to only get hydrogen. By integrated system, we try to integrate methanogenesis, photosynthesis as well as CES. We'll be able to get three forms of products. In this system, we are not separating VFA, but using for methanogenesis followed by photosynthesis. So by able to convert into three forms of products. Apart from 90% of CO thermal efficiency. So now any biological process, I would like to say that when we cannot consider it as sustainable because we are using biological system and waste, but we have to assess its sustainability by life cycle strategies. So we try to use LCA strategy here, and we try to understand the hydrogen production process by taking into consideration a standalone process where hydrogen is only the product by this system boundary where food waste, sodium, chemicals which is used to buffer water and electricity are feedstock. Hydrogen is a major product apart from CO, carbon dioxide, organic fertilizer. of and as well as estrogenic effluent. The system boundary for biorefinery system, wherein all inputs are here, but different unit operations, which I explained earlier, are there. Apart from number of products, apart from hydrogen CO2, we get oxygen because of photosynthesis, and also methane with anaerobic digestion, and treated water for recycling, and biomass for photosynthesis, as well as partially estrogenic effluent for organic fertilizer. So multiple resource recoveries. These are the two system boundaries. Try to put it in LCA, wherein you can see the 16 dam impact categories by ISO 1404 framework. So standalone process is producing near to 4.1 kg of CO2 equivalents per kg of hydrogen. But this is a negative because the system, the CO2 is producing biogenic in nature. LCA considered biogenic CO2 as not a harmful CO2 as fossil based. However, taking into consideration it as a conventional CO2, 1.21, when we are integrating biorefinery, you can see significant reduction of CO2 emissions from 4.21 to 1.03. So by way of integration, we can reduce greenhouse gas emissions. Apart from that, when we look compared with four damage categories: human health, ecosystem, climate change, and resources. This is a standalone system, and compared with the existing hydrogen production process, wherein human health was reasonably good compared to one of the process, M2, but ecosystem services are bad. It is bad as much as M2. However, climate change, there is negative emissions. Resources, we are only using waste, no fossil base, so it is also negative side. But when we compare with the biorefinery concept, you can see human health has reduced significantly, and a similar ecosystem quality, because we are not sending any waste into the system, and climate change also reduced significantly from 0.4 to 4 to 1.1, and resources also reduced. So there is overall sustainability with less environmental damage. So compared, just for comparison sake, so the four process which is conventionally being used, and the government is looking into green hydrogen by electrolysis. When the electrolysis process nearly consume 62 kg of CO2 equivalents for 1 kg of hydrogen production. When we go with the coal-based system, our coal-based system will have energy mix of 80% of fossil and 20 renewable. But when we are replacing fossil-based energy mix with the solar panel, significant reduction, 1 kg of hydrogen, we can able to produce with 2.7 kg CO2, which researchers are still trying to reduce this, then it can become green hydrogen. So likewise, green biological also, we are able to get near to 1.2 kg CO2 per acre. So based on this, we are setting up a pilot plant, not pilot plant, prototype plant with funding of Department of Science and Technology, with a capacity of 150 kiloliters per day of green hydrogen. Basically, this contains 3 modules, wherein module 1 represents biological process, different biological process as feed preparation, pre-treatment, estrogenic process, methylogenic process, as well as anoxic and microalgae cultivation. So the outlet from hydrogen reactor, liquid outlet will go to module 2, wherein fatty acids will be separated here, and the gaseous emissions will go for module 3, where it is subjected to separation of hydrogen as well as CO2, and subsequently storing. So this based on a pressure swing absorption. So we'll be able to capture hydrogen, and by methylogenic process methane, we can give hydrogen as well as HCNG. Apart from fatty acids, the residual fraction of fatty acids which are not able to capture will go for methylogenic process. Treated water again go for closed loop operation. This we are trying now preparing DPR for scaling up. So to conclude, I would like to say there is a lot of scope, particularly with respect to refinery, biorefinery kinds of system, because India has huge amount of waste, and there is always increase in CSIR rate, particularly for bio-based materials in coming days because of its sustainable nature. However, this required significant interventions, particularly with respect to policy and with respect to technologies. Upgradation is also required, and techno-economic analysis has to be done. And market has to accept this product in place of low cost fossils. So the movement is towards bioeconomy and circular economy, as well as using biorefineries, and people are considering waste is not a waste and an important feedstock. And future will be with these kind of interventions, we can reduce our requirement of fossils for our material requirement. So, however, these required the interdisciplinary nature of research, then that will be fruitful. So, I would like to thank my students whose work I have presented here. And funding from different funding agencies, starting with RBT, who funded us first hydrogen reactor, hydrogen project, as well as MFC, and MNRE for pilot plant, and DST now for prototype plant, as well as the CSR for waste biorefinery facility. Thank you very much for your kind attention. I'll be happy to take questions.
Thank you so much, sir, for this